All right, very good afternoon, everyone. I have already uploaded this lecture 6, Mendelian Genetics, in the YouTube. I hope you got opportunity to visit this lecture and at least get some idea for Mendelian genetics. Nevertheless, I'm going to recap some of the concepts, not all the slides, once more, and some of the Q&A we will take, followed by the quiz from the same lecture on genetics. So outline for this presentation is that some of the concept of inheritance from Mendelian genetics. I'm going to recap that. We'll give you some examples of Mendelian genetics and then move on to some of the probability rules, how mathematical equations and rules can be applied to find the same findings what Mendel arrived at. I'll let you first make more familiar with some of the terminologies. And these are very basic terminologies, which is, I think, good for you to recall, because many of the time you are going to see these terms in the different definitions. So first is the genotype and phenotype. What is our appearance? That comes under phenotype, whereas what is the genetic composition? That comes under genotype. So while we are going to talk about this cross of Mendelian inheritance, looking at the pea flowers, but just for the time being, looking at the appearance of the flowers, if you look at here, we are seeing a purple colored flower and we are looking at the white colored flower. What we see here, two different type of phenotypes. One is purple, one is white. However, if we look at more closely, some of the purple colored flowers have the gametes, which is capital P, and some of the purple flowers have capital P, small p. So now we can dissect out these terminologies more and look into the terms like homozygous and heterozygous. What we see, the capital P or a small p, these are homozygous, whereas capital P, small p, these are heterozygous. So just by looking at these flowers, if we want to now look at just the phenotypic ratio, what we saw, 3 of the purple flower and 1 of the white flower, which is known as the phenotypic ratio, 3:1. And if we look at their genetic composition or the gametes, it looks like PP, capital PP, small pp, these are homozygous, whereas there are 2 heterozygous, capital P, small p. So genetic ratio for the genotype is 1:2:1. When we have different alleles, those are the heterozygous, as I explained here, capital P, small p. When we have the same alleles, these are the homozygous or the identical alleles. So these terminologies are going to be used many times. Of course, we'll talk about this cross subsequently, but I just thought to give you some definition so that you are clear about these terms. Also, we have discussed about genes, and we have talked about by now in the DNA tools that we have now ability to look at the gene sequences, the nucleotide sequences, which was not possible earlier. At the time of Mendelian science, he was only thinking that, okay, there are some stretch of sequences which are providing some information which is inherited from one to the next generation, and those are known as heritable factors or units. And those later on only we know that these are known as genes, and there is a nucleotide sequence which can be sequenced using next generation sequencing technologies. Now the same gene, there are 2 copies comes which is inherited from both the parents, and therefore each organism harbors 2 copies of the genes which are known as alleles. In this case of the flower which we are talking, now the gene which is giving the coloration, that is either purple flower or the white flower, there are 2 alleles which are present on different chromosomes coming from the pair of homologous chromosomes from 2 parents. So now the question comes that, okay, why one of the flower looks like purple colored and one flower was white colored, and what makes it appear dominant and other becomes recessive trait? If you look at their biochemical composition and now more nucleotide composition, because as I said, now we have ability to look at these sequences and look at the next generation sequencing and whether type of technologies are available by now. But even the basic genome sequencing techniques can tell you the possible sequences for those genes. So looking at that, what it appears here, that if we look at this nucleotide sequence, CTAAA, TCGGT, this is sequence which is responsible for the flower coloration in the pea plant, and that gives either purple color or the white color. But this exact sequence gives rise to the purple coloration because, like, as you know, that from the 3 nucleotide, it generates the amino acids, which is going to generate polypeptide sequence followed by the protein synthesis. enzymes are proteins. Some proteins with enzymatic activities are also being produced. So this particular sequence generates series of reaction which generates purple pigment and with some enzymatic reaction, and therefore the flower now appears purple color. Whereas the same sequence, if you look at here, same nucleotide sequence, only one change is here. C has been replaced with A. And as a result, now the same triplet codon, like when you look at these three, is not going to give rise to the same protein or same enzyme. So therefore, what you may realize that it's not going to produce the same enzyme and not going to give rise to the same color or pigment, what we saw in the earlier example. So therefore, what we can conclude that there is something specific in this sequence which is generating these enzymes, and therefore we can see some color production happening, and that's why it is termed as a dominant trait. Of course, it can always be subjective that what is dominant, what is recessive. Nevertheless, here there is some specific transcription and translation activities have happened. As a result, some proteins being synthesized, and now we have some unique product being formed, like purple colored product being formed, which is giving rise to purple color. Like that, we have many phenomena which we are going to talk, and just keep in mind that there is something specific happening, at least in the dominant traits. Something is more getting synthesized or more products being formed. As a result, some superior traits of the same phenomenon being observed. And that's why we call them as the dominant, because many of you have asked some questions along these lines in the Google form. And I hope some of these things are clarifying to you. So this is the difference in dominant and recessive, purple versus white colored. And now we have all the information at the sequence level. So thinking about this, given that this class is coming from all the students of engineering background and the aspirant to become engineers, all of you have interest in physics, chemistry, mathematics more than the biology. So Mendel fits very well here with the example that he was also of the similar interest of keen studies in physics and chemistry at University of Vienna. And he, of course, did not want to become a biologist. It just happened that he became the father of genetics, but he never did the experiments for the sake of doing the experiment in doing a research. He was just, a teacher in the monastery, and he was teaching over there, and in the garden, he was growing the pea plant. And as a result, he, just out of curiosity, he started doing plant breeding much more meticulously. With his mathematical inclination, he was able to look into how best he could try to count those seeds and observe them one to next generation. And that was actually quite remarkable, because that gave him ability to come up with some common factors which he saw, which is observed in different type of properties. And that led him to the generation of Mendelian rules of inheritance. Let me just show you this small clip, which will intrigue your interest more in this particular Mendelian's biography.
Effect, and with a mathematics professor named von Ettinghausen, who had developed this theory called the combination theory, the idea that any group of objects, man-made or natural, the relationship among them can be expressed in some sort of a mathematical expression, some sort of a combination. These kind of ways of looking at the world really informed Mendel's experimentation when he got back to the monastery, and really helped him figure out a way to look at his peas and his results in a whole different way, in a mathematical way, instead of just an impressionistic way.
Mendel had limited funds, so he was fortunate to find quarters near St. Elizabeth's Hospital in a building supervised by the nuns. At the university, Mendel came into contact with some of the leading scientists of the time, including Carl Nageli, a renowned plant expert, and with whom Mendel later corresponded for many years.
Gregor Mendel studied a number of subjects here at the University of Vienna. One was physics with Doppler, for instance. Another was botany with Unger, and many other subjects such as mathematics, astronomy.
What Mendel learned from these men would shape his future: the mathematical analysis of natural events, the statistical principles of meteorology, the study of plants from a historical perspective, and the development of hybrids.
He returned to Brno in 1853 to continue his high school teaching, even though he still hadn't passed the required examination. On his second attempt, he had a nervous collapse and became too ill to go on. While he continued teaching, Mendel began a series of experiments in plant breeding that were aimed at answering one of the most important questions in biology: what is inherited and how? The answers that Mendel obtained from his plants became the foundation of the new science of genetics. It had long been assumed that plant reproduction just happened. Mendel didn't accept this answer, but pursued an idea introduced by Carolus Linnaeus: plants have sex. When the subject came up in his classes, Mendel described it in some detail, accompanied by classroom giggling. "Don't be silly," Mendel would say. "These are natural things."
One of the few trips abroad that Mendel took was to the London exhibition in 1862. He was part of a delegation from the town of Brno who had put together an exhibit on crystallography. It's interesting to think of Mendel having really studied crystallography right around this time when he was also growing his peas and getting ready for his description of inheritance, because crystals, sort of like genes, follow a particular pattern when they pass things on from one crystal to the next.
Mendel used peas in his study of plant reproduction. He chose the genus Pisum because it produces fertile hybrids and it is easy to grow. He studied peas to determine how traits were passed from one generation to the next. Others had observed these traits. Mendel was to draw extraordinary new conclusions. Mendel discovered three fundamental laws of genetics. First, each trait is determined by a specific element. Today, we call them genes. One gene determines, for example, a person's hair color. Another determines eye color. Second, each characteristic or gene is inherited separately without affecting the others. For example, hair color is inherited separately from eye color. Third, each inherited characteristic is determined by the intersection of two genes, one from each parent, and one is always dominant over the other. The dominant trait will be passed on from one parent. A recessive trait must be passed from both parents.
He did 6 other experiments on peas using different traits, again with remarkably similar results. He had laid the foundation for the modern science of genetics, defining the fundamental rules for dominant and recessive traits that apply to all plants and animals.
In 1865, the Society for the Study of Natural Sciences in Brno invited Mendel to present a lecture on his experiments. Mendel was not a well-known scientist, nor was he associated with a well-known institution.
Nobody said a word when he was finished. There were numbers everywhere because what Mendel did was counted up all these peas, looked at their different traits, and came up with ratios and mathematical equations that would express the relationships among the different traits. Clear effect.
All right. So I hope that just conveyed you that, Mendel was really lucky in some way that, whatever observations he made, those resulted into some very solid background from the different properties of pea plant. And, as we go along, you will see there are different properties in pea, which is all giving similar results to him. And that's why his model became very robust, and he was, considered that, now these rules can be taken forward. Otherwise, he was not very well-known scientist, as you've just heard, and neither he was getting a lot of prominence and acceptability of his results. So question comes that how to define heredity, and there has been different school of thought for how one could define this. For instance, like, there's a blending hypothesis, which says that these genetic materials are contributed by two parents, like you can mix different type of paint colors, which blends and gives rise to different color. Or there's a particulate hypothesis, which tells that, the idea of the gene comes forward from this hypothesis, that if you can take an example of collection of cards, if you're playing the cards, and the different players in the card, so likewise from the parents moving the information to their offspring, son, daughter, or to the grandson, daughter, all of these could be shuffled, and that is one could find some pattern of information which led to the ideas of gene. So keep in mind some of these hypothetical names, the hypothesis from blending and particulate hypothesis. And this one is more acceptable as the idea for the genes, how it works. As I mentioned, the Mendel was quite lucky in choosing the pea plants for his experiment. Think about, various type of model system, which on and off we have been talking. We discussed about various type of organism I showed you, like C. elegans, Saccharomyces cerevisiae yeast, or looking at some bacteria. In fact, even the sometimes monkey can be a good model. And pea plant is not so well known model. In the plant, there is Arabidopsis thaliana, which is a better model. Model means like these organisms have been understood quite well. Their sequences have been analyzed well, and they grow very fast, and there is a faster generation rate. So therefore, the experiments are easier to done. So many scientists who work in the plant area, they choose Arabidopsis thaliana, one of the mustard family plant, because this plant sequence is known. Its generation time is very fast. A lot of seeds can be generated, and you can do many experiments in that. So Mendel, with some rationale, he chose pea plant because he found that there are many variations available in pea plants. He could see some are tall, some are dwarf, some are having white colored, some are purple colored, and like that many characteristics he could see. So he chose this plant. Also, it was easy to do the breeding in this pea plant. Of course, it's a good plant to have in your garden. And then he was able to separate the pollen and the egg bearing organs so that there is no cross pollination occurring, and he could be more confident about the results. Likewise, he could see that there is a short generation time. It means the pea plant can really, does not take so long time to grow. All of these led him to choose the pea plant. And one thing which came forward based on his experiment, that why he became very successful, because he understood that, okay, there is some difference. Even he sees the plant look like tall plant, but that tall trait may not always remain tall. So it may segregate in some generation. That's what he was observing. And likewise, a purple flower, it may further segregate into purple and white flower. So to do an experiment of the the crossing, he made sure that he's choosing true breeding varieties. And please do pay attention to some of these the terminology which I'm speaking. I'm actually now repeating myself because it's already in the lecture I have given you. But still, I'm just emphasizing these terms. You should try to read a lot of questions which are going to come based on many of these concepts which I'm trying to re-emphasize. So true breeding, it means like he's going to take only purple flowered plant. It's going to do cross and keep doing cross till he always see only purple flower plant in subsequent generation, and take seeds from those. And then only he will choose those seeds from the purple flower plant. Likewise, he will do cross of the white flower plant. Subsequent generation, when he sees only white flower plants are coming, then take the seeds from the white flower plant. So now the cross could be made from the purple versus white, but both are true breeding varieties. It means they are not going to segregate into purple and white or white and purple or tall trait, tall and dwarf like that. They will be consistent with that trait. This is what is known as the true breeding variety. Some of you have also asked to explain this question or the concept. So many characteristics which Mendel was studying, and I must say that there has been a lot of criticism sometime or question sometime that why Mendel was very lucky of doing these experiments. Many people are quite envious because Mendel was very lucky to find many traits which also showed similar phenomenon. Same dominant and recessive trait was observed in at least 7 properties of pea. For example, flower color, purple versus white, flower position, axial versus terminal, seed color, seed could be yellow colored or green colored, seed shape could be round or wrinkled. It could be pod shape, which you eat this or you buy from the shop. You see these are sometimes inflated, sometimes constricted. Pod colors, green or yellow in color, or the stem length could be tall or dwarf. So what you see, like there are 7 contrasting characteristics which could be following the same type of traits, and those characteristic variants are known as the traits. So Mendel, from his observation, he determined that there is some commonality and some units or the factors which are passing the information from the parents to the offsprings, and individuals are inheriting at least 2 alleles from that one such unit or gene from their parents, and that trait may show up in the individual or it may not show up, but it may show up in the next generation. So therefore, some traits could be hidden in that way. And based on these observations and some experiment, then he proposed law of segregation. In law of segregation, the experiment which he did on the pea flower was essentially the true breeding purple flower and true breeding white flowers were chosen. True breeding means it's going to always have only capital P gametes, and white flower can be small p. Again, feel free to write any nomination, whatever you want to write. Whenever you have been given a question based on the genetics cross, it can be just X, Y, or A, B, whatever you want to do. So there is no set nomenclature why you have to write only that. So in this case, from the parent generation or the P generation, the gametes produce this capital P or small p. And now what you see in the first generation, F1 generation, or the filial generation, there you're going to see the heterozygotes or capital P, small p, heterozygote purple flowers. And these gametes now, when they were further crossed, you can see this is known as the Punnett Square, like a square which you play sometime game. Now you got the gametes coming from the sperm or the egg, which is capital P small p and capital P small p. These are the heterozygotes coming from the F1 generation. After doing this cross, he observed that 3 flowers looked at phenotypic wise purple color, whereas 1 flower was white in color. However, as we just mentioned earlier, that one of them is actually homozygous dominant, and now by now I'm sure terminologies are clear to you. Homozygous because both are same, homozygous dominant because it is going to show the dominant trait, and heterozygous dominant. So all three of them are showing dominant trait, but one is homozygous, 2 are heterozygous. This is recessive in trait and is still dominant. It's still the homozygous. Sorry, it was not dominant. It's recessive, but it is homozygous in nature. So these are the kind of thing which he observed from this experiment. However, as I mentioned that when looking at the purple flower, you cannot be sure that is that going to be homozygous or heterozygous. How to do that to ensure that the purple flower, or for the same matter, when you look at the inflated versus constricted or the tall versus dwarf, whether those characteristics are going to be homozygous or heterozygous. So to determine that, the experiment which we can do is known as test cross, where you take the homozygous recessive. This is always fixed. You are going to take your homozygous recessive, which you are going to cross with the unknown, which is not very clear what should be genetic composition or the gamete composition of this flower. So now what you see here, you're always having like a small p, small p, small p, small p in both the crosses. Whereas the X, which is coming from the purple flower, could be capital P or could be capital P small p. It's good idea for the genetics experiment and the crosses that if you can do these things yourself in your notebook, please do that. When we are in the classroom, that we really ensure that everybody is doing it, and it becomes very comfortable for you to then realize these are very simple things which can be done very easily and has a lot of logic into that. So you don't have to memorize anything here. It will be very straightforward. Please do that if you have not done by now, and do this cross yourself. For example, the purple flower, as we have just seen earlier, it could have 2 possibilities. It may have PP, capital PP, or capital P small p. Now, to determine that what could be composition of this gamete, one is doing the test cross, where always you are going to take homozygous recessive small p. And based on these experiments, what you see, like one possibility is that all the offsprings are purple. And in that situation, then the X are having the capital P, capital B, capital P, which is homozygous dominant. Whereas it could be 1:1 ratio also the result, where it could be capital P and small p. So these are the things which can be easily deduced by looking at test cross. And these are the things which we are going to ask you some questions at different time, that how these, and for an unknown characteristic, how you are going to determine the traits and which type of test you can do and what will be the partner with which you are going to do the cross. And if we give you observation, like if you see 1:1 ratio or 50% offsprings with the variation of the traits, then what can be that possibility? And you have to look into this cross and come up with, okay, what can be capital P or small p in this case? And if it is 100% or all 1:1 ratio, then it is going to be capital P, capital P. So very straightforward. Questions will be based on this very simplified way, but you have to do the cross. So please start doing that. So far, what we have discussed, that in Mendel's law of segregation, he suggested that genes have alternative forms or the alleles. 2 alleles, they are going to inherit from the parents, which may still further segregate during the gamete formation in the subsequent generation. He showed that in the F2 generation, the phenotypic ratio is 3:1, whereas the genotypic ratio is 1:2:1 when we are looking at monohybrid cross or one property at a time, right? So just keep in mind, whatever we are talking in the law of segregation is based on monohybrids or one property at a time. And to determine the individual characteristics of these gametes, a test cross can be made to decipher the genotype. So these are quite a straightforward experiment. Mendel was quite happy and quite lucky that he had 7 characteristics to prove his observations in P plant. But now comes a little bit more complexity, that given that P has many characteristics, are they going to be assorted together in the dependent manner, or they are assorted independently? And to test out this possibility, then Mendel thought, can we choose the 2 variations? One is seed color and one is seed shape. Seed could be yellow in color or green in color, right? And likewise, it could also be round in shape or wrinkled in shape. So now if we look at 2 properties at a time, so keep in mind, independent assortment law is going to look into dihybrid cross. It means we are looking at 2 properties at a time, whereas the law of segregation was looking at 1 property at a time, and that is known as monohybrid cross. I hope you started making this comparison in your mind or a table, which will help you to think about various situations which we will ask in the questions. So here, look at this seed, which is now yellow in color and round in shape, whereas this particular seed is green in color but wrinkled in shape. So now we are looking at 2 different properties. Are these properties inherited together or they're independent of each other? It is not known. So therefore, Mendel thought to do this experiment where now he did cross with this particular seed, which is yellow and round, and this seed, which is green and wrinkled. And what he found that in the first generation, like earlier experiments, he see only 1 characteristic being dominated. And now he see this is the heterozygote being formed. Further, it could have been 2 possibilities. Again, now what I'm showing is Mendel did not know that time. Is that going to independently assort or going to be dependent on the in the next generation? So if Mendel would have got result like this, then which is the phenotypic ratio of this yellow round versus green wrinkled seeds, 3:1, it means these properties are dependent on each other and they are going to assort dependent manner. However, what he saw was something different than his previous experiments. He now saw phenotypic ratios are 9:3:3:1, which means it is quite different than what he could see in the monohybrid crosses. More properties being observed here. And based on this, then he formulated that these are probably independently assorting. It means these properties are not inherited together and these are independently assorting. This phenotypic ratio is not always 9:3:3:1. This 3 could be 3.1 and 3.14 like that, but approximately 9:3:3:1 ratio was observed. How can you do these experiments? Of course, you can think about either doing the Punnett Square or you can use the probability rules. So since we are saying there are 2 different properties and both are inherited independently, so you can try to simplify this problem into 2 simple monohybrid cross, for example, seed color and seed shape. If you do the cross of seed color and look at the possible gametes like 1/4 capital Y, 1/4 small y, and half of the capital Y, small y, like earlier what we did the cross. Same goes to seed shape as well, and you can see this seed shape gametes. Now you can utilize this information to look into the further crosses and see like what could be possibilities of different type of gametes being formed. But if you want, you can use the much simpler way of looking at probability rules and say, okay, since I'm going to look at 2 properties at the same time, if I take these 3 gametes, 1/4 of the capital R, 1/4 of the small r, and half of the heterozygote is capital R, small r, and each one of those I do the cross with the 3 gametes from my seed colors, which is 1/4 of capital Y, 1/4 of a small y, and half of the capital Y, small y. And then you can do the cross and see, okay, 1/16 of capital R, capital Y is found. So now our questions which will be asked to you will be something like this. We give you some gamete and we'll ask you what could be probabilities of these F2 genotypes. And if you can just simply do this type of math, you can come up with any type of gamete composition. You don't have to work on this type of a scary image like this. Of course, one could look into the entire cross and the Punnett Square checkerboard and try to come up with all the gametes and possible crosses, which will give rise to these gametes formation. But you can rather look into much more simplified numerical ways of deriving all these gamete composition, which will give rise to what is the probabilities of these gametes to be formed in the dihybrid cross, because dihybrid cross is slightly more difficult. Here we are talking about two properties coming together and still they're independently assorted. So based on this, so far what we learned that Mendel formulated his second law of inheritance by studying two characteristics at the same time. He stated that these genes are assorted independently because his observation did not lead him to the 3:1 ratio, right? Think about how elegantly, just by looking at the ratios of the pea plants and seeds, he was able to make this conclusion, which still remains valid after centuries. Each pair of these alleles segregate independently during the gamete formation, and his crossing experiment in the dihybrid crosses resulted into phenotypic ratios of 9:3:3:1 ratio. So far, what we have discussed, there are two laws, where we have talked about law of segregation based on monohybrid cross, law of independent assortment based on two different properties of the dihybrid cross. The phenotypic ratio was 3:1. Here, the phenotypic ratio is 9:3:3:1. All right, so that just conclude the first part of it. I'll just recap this concept in a nice animation here. So let's just watch this animation.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett Square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase Y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3:1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
All right, so after looking at those crosses, probably you're more refreshed with this animation. And let us now discuss about some of the examples of Mendelian genetics. So what we are looking at is so far is complete dominance, which you have seen in the heterozygote, there is some characteristics which are hidden, and one could look into how they get segregated, but still they are going to show up in the next generation. Now we have incomplete dominance, which is a new terminology which we are introducing you right now. In addition to that, the purple versus white, if you do the cross, you may not observe either purple flower or the white flower, and some different color shows up. And that's where that comes incomplete dominance, or it means both the partners are probably equally dominant. And now what you see is the different trait being appeared, right? So that is incomplete dominance. Or co-dominance, when the two dominant alleles affect the same phenotype separately, but that could be distinguishable way. And we are going to show you the examples of blood grouping in this particular terminology. So let's look at this interesting plant, a red snapdragon. Here the flowers are either red colored or white colored. What you see like in the parent generation, if you do the cross, here let's say we denote our gametes as the C capital R, C capital R, and the white flowers as C capital W, C capital W. What you see here, R and W, which is both are capital, and resulting into new type of gamete formation in the F1, which results into pink colored flower. Now this pink colored flower, the seeds from that, when you take and do the further cross of RW and RW, what you see is the characteristics are further segregated, and now you see a red, a white, and pink. The ratio turned out to be here 1:2:1. I hope you recall that we had the phenotypic ratio in the F2 generation, which was 3:1, right? Now what we see is the 1:2:1 ratio. Earlier, we have seen the genotypic ratio was 1:2:1. So here you have to keep in mind that, what we are looking at, that phenotype as well as genotype, they're both differing. Whereas in the case of, when we talked earlier, we have seen it was 3:1 and 1:2:1. Here it remains 1:2:1. So these are the kind of thing which can give you some hint if we are asking you about complete dominance or incomplete dominance, and I'm sure you'll be able to pick up with this example. So other interesting example which we see in nature, which happens routinely, is essentially we see like, some child are born with the white hair, which is a lack of pigmentation. There is some protein, some enzyme, which is not, able to make here. And as a result, you don't see the, pigment being synthesized. That's all it happens. And that is being caused because of the homozygous AA recessive trait. So if you look at this parent, which are normal parents with the heterozygous AA, capital A, small a, and now from their sperm and eggs, you do this cross. And what you will observe here, capital AA, capital A, small a, heterozygote, capital A, small a, and small a, small a. So now one is normal child, two are carriers, and one is albino. Albino is this particular phenomenon. So if in this family, let's say only one child is born, or two childs are born, or three children are there, so they will not show up any albino trait. However, if there are 4 children born, there is a possibility of this albino trait also to be seen. So therefore, you don't see these traits appearing all the time in a very common way in nature, but they are getting inherited from one to next generation. And only when these homozygous AA individuals are born, they are going to show this type of coloration, no coloration or lack of pigmentation. But more interestingly, what we observe is our own blood grouping, right? And which is ABO blood grouping. Now here, multiple alleles are partnering to show some phenomenon. What we know is this is red blood cell, right? RBC, red blood corpuscles. On top of these RBCs, there are some antigens, which is making them different. And these antigens are some sugar molecules, which is carbohydrate A or carbohydrate B, right? So now if we look at their allele, what we have for the gene, which is causing this phenomenon, if we denote that with I, so we got I capital A, which is one of the sugar type present on our blood, or I capital B, which is another sugar type present on the blood, or there is no sugar present, that is the small I. So if we look at our how we can provide notation to the blood grouping, genotypic and phenotypic wise. So phenotype wise, we have like this particular RBC on which we have triangle carbohydrate sugars that is giving rise to A blood group. Now we got another one, which is the circular one giving rise to B. Both of them are present, that is AB. And now when we have none of the alleles are present, that is O blood group. These are phenotype. However, from the genotypic wise, how you can look at I capital AA or I capital A small I. Both of them are going to give rise to A blood group. Likewise, I capital B or I capital B small I giving rise to B blood group. Both are dominant, I capital A and I capital B, that is giving rise to AB blood group, and both are absent. You can show them with a small I. Please do try to practice these notation. It is going to be used multiple times for the crossing. When you have the A blood group, both are possibilities. Whereas when you have AB blood group, only this is a possibility. When you have O blood group, only this is a possibility, right? How to determine the blood group is a very interesting experiment, which I think you can first get a glimpse from this video made from Amrita University, and I'll continue from there.
An adult human has about 4 to 6 liters of blood circulating in the body. Blood consists of several types of cells, including white blood corpuscles or WBC and red blood corpuscles or RBC floating around in fluid called plasma. There are certain protein molecules called antigens located on the surface of the red blood cells and antibodies which are in the blood plasma. The differences in human blood are due to the presence or absence of these antigens and antibodies. Individuals have different types and combinations of these molecules. According to the ABO blood grouping system discovered by Karl Landster, there are 4 types of blood groups: blood group A, blood group B, blood group AB, and blood group O. Rh or rhesus factor is found on the RBC surface in most people. Like A and B, this is also an antigen, and those who have these antigen are called Rh positive. Those who lack this antigen on the surface of RBCs are called Rh negative. To conduct the experiment, we require 3 monoclonal antibodies, anti-A, anti-B, and monoclonal D, kept in an icebox. Lancets, spirit, cotton balls, toothpick, and glass slide. Procedure. Swab the finger with cotton balls dipped in a spirit and take blood sample using a lancet as shown. Take 3 separate drops of blood on a glass slide. Place a cotton ball on the finger to stop the blood flow. Pour 1 drop each of anti-A, anti-B, and monoclonal D on each drop of blood respectively. Mix the monoclonal antibodies with the blood sample properly using a toothpick. Make sure you take a new toothpick for mixing each drop. When RBCs carrying one or both antigens are exposed to the corresponding antibodies, they agglutinate, that is, they clump together. For example, in the slide, agglutination occurred on the first and the third drops, which corresponds to antigen A and Rh factor. So the blood group is A positive. Thus, we can identify the blood group by noticing the agglutination on the corresponding drops.
All right, it seems Alok had some very interesting burning question, but Alok, just if you could hold on your curiosity for 2 minutes more, we will be with you. So now just concluding this ABO blood grouping part, what you just saw, like, by doing some simple reaction, which is antigen antibody reaction, these antibodies actually are generated against the epitope of antigen. And in this case, antibodies against A or B, or as well as the Rh factor for the D. So there are 3 antibodies which is being used for these reactions to know. And if you just focus into this box right now, what we are looking at, if you are adding the antibody A in a given unknown blood group. So let's say you do not know your blood group, and 3 drops of the blood is kept on the slide, glass slide, and 1 drop you are adding antibody A, other drop you're adding antibody B, and 3rd drop you're adding antibody D. So what you will observe, like, if there is agglutination happening, which means antigen antibody reaction is happening, and that result into the precipitation of the protein. So that is giving rise to idea that this probably is going to be blood group A, whereas the B is not showing any reaction here. So you can, based on this data, you can only say that this is the blood group A. But now when antibody D is reacting here, it says that it is going to be A positive. This blood group is A negative, right? So now if you move on to the next one, you will see antibody B is reacting here, whereas antibody A did not show any reaction. And now the antibody D is showing reaction, so this became B positive. If you move further, you will see, okay, both antibody A and antibody B are showing reaction, whereas antibody D is showing reaction in this case, so that became AB positive, and this became AB negative. Further, now we see there is no antibody reactions are happening with A or B, so that is giving rise to O blood group, And now we are looking at the O positive or O negative based on the rhesus factor. So let's move on to show you this experiment live from the lab, just so that you will better expose. And when we were teaching the course in the classroom, we do show some of the experiment with the unknown sample students, like do want to test their own blood group to see that really works. And it was very interesting to see that how specific these reactions are. And given that now when you go to any medical center, any medical hospital or path lab, when you go for your blood test, in 2 minutes time, you will know that what is your blood group. And that is because of these very standard reagents and tests and knowledge what we have against these antigens and these antigen antibody reactions. And therefore biology is very logical, and it actually works always very robustly, right? So I'm requesting now Meher, a TA from the lab, to show the experiment live and just give you some idea how you can predict the blood grouping. And you have to make some idea and prediction that what is that blood group which we are showing you. So Alok, you can talk after this. So just let's finish the blood grouping, then we can talk. Go ahead, Meher.
Thank you, sir. Hello, everyone. So now we'll show you some steps that are involved for the identification of blood group through ABA blood group system. So here we have different material which is required for this experiment. So we have this glass slide, and we have the antibodies from the kit, including antibody A, antibody B, and antibody D. And from the same, we have taken few aliquots that we'll use. So first step is, firstly, we have to sample the blood. So firstly, we'll wipe the finger, and then we'll, using the lancet, we'll take the blood sample. So as being shown here, so gently just pricking with the lancet, we have to now place 3 small drops onto the slides for 3 different immune reactions. So 3 drops on the slide are placed, and now different antibodies will will put on it. So such as now putting the antibody A. So depending upon the results, we'll conclude finally which blood group it was. And now antibody B. And similarly antibody D. So now using a clean tape, we can just mix them, blood and the antibody, to know where this agglutination is happening. So this was the anti-A. Now you can guess in which of the reaction agglutination is happening, and you can identify the blood group. And now mixing the anti-D reaction. So now you all can guess which blood group it was. So I can see some agglutination in the first one, and there is no agglutination in the second one against the anti-D, but there is agglutination against the anti-D. So accordingly, we can identify the blood group. Thank you.
All right, thank you, Meher. So I'll just, so student, I think you have started making guesses, which I'm sure you're right. Since you saw the agglutination in A, that was A blood group, and given that you got to see that there was something happening in D, antibody D reaction, that is known as the A positive. So I think that now gives you a very good idea that if you are given some results, how you can interpret from that and what kind of conclusion you can make from that. So just to summarize, I think today what we have discussed, we look at some of the Mendelian concept of inheritance, experiments which he did very elegantly, very robustly on the, with the breeding, resulted into many concepts which remains even valid till date, which resulted into law of segregation and law of independent assortment. We have also discussed various examples where some variations can be seen. And of course, many of the hereditary disorders, some of them do obey Mendelian rules. Some of them also are more complicated than that. And of course, we've also talked about how you can solve some of the problem based on the rules of probability. Some participants are really curious to ask some questions, but let me give some preference to the questions from the Google form chat for the students who have asked questions before you. So some questions were asked based on the, what is recessive, what is dominant, and especially some confusions and questions are toward the, incomplete dominance, right? And that's where we see 1:2:1 ratio being observed for the phenotype and genotype. And there is no recessive or dominant alleles here. Both of them are showing contribution which is equal, and therefore we see them as an incomplete dominance. Another question interesting being asked is, how can Mendelian genetics be used for the cancer analysis? Cancer is a very complex problem, as we have been discussing. There is possibility for the genes to show the heritable traits. At the same time, there are few things which governs beyond that, which is including the environmental impact, as well as many mutation which occurs, which make it really complicated to always follow Mendelian way of inheritance. So therefore, we cannot say it's only follows Mendelian heritable trait, but actually it's much beyond and much more complex than that. A few thoughts and questions surrounding to these topics, but I think by now you are clear. Complete dominance, mainly when we talked about Mendel's monohybrid crosses, and we look at one trait was only being inherited dominantly. Incomplete dominance, we gave this example of purple flower of the snapdragon, and co-dominance we discussed with the blood group, when you can just see like both A and B are equally participating in this phenotype. So co-dominance, again, some questions being asked that, how this works. Now this is, you can really look at the genotype notation about them only in I capital A, I capital B, which is AB blood group. Here you see like, both of them are equally participating, and there are other possibilities of allele, which we have discussed earlier. True breeding, I have already discussed that. Many of you were curious to know what true breeding means and how Mendel's experiment actually led to the right conclusions and why the true breeding came into the play. So given that if this is only going to be capital P, and there's going to be only small p, therefore the crosses are going to make sense. But if this is going to be capital P, small p, and this is going to be small p, this is going to change. So therefore the true breeding was important to ensure that whatever he's seeing is getting inherited subsequent to generations, and he could see those seeds obtained from those plants. What makes an allele dominant or recessive? Can a dominant allele become recessive later on, or as it passed over generation? Very interesting question. I think one of you have asked. I have been explaining this, that, okay, like one is dominant characteristic, one is recessive, but dominant is because it is able to produce some pigment or some enzyme, which leads to the pigment formation. Therefore, we are saying it is the dominant trait. It means during the transcription and translation, there is some superior trait being observed, and that is resulting into this particular phenomenon being said as a dominant. However, it may happen that some mutation occurs, and therefore that product does not form subsequently in the generation, and therefore this phenomenon may also get reversed. So you are thinking ahead of time, and I think that's good that it could actually be vice versa as well subsequent to generations, but in general it may not change very easily. Law of independent assortment has any bearing with crossing over and meiosis? Yes, I think what we learned in the meiosis, when the gametes are showing the recombination, that is quite linked here when we are looking at during the meiosis, chromosomes are separated into multiple gametes, and likewise we see here in the law of independent assortment these characteristics which are being inherited, but still they are forming their separate entities or separate characteristics. I think that's pretty much it for various, the most of the question which you have asked. I think we have tried to group them so that to save the time. And you should read this particular chapter 14th from Campbell book, which will be coming to today's quiz, but more importantly, it will keep coming back to you. And we will have some significant proportion of the genetics as we go along in the mid-sem as well. So you should be thinking that, how best to learn these crosses, and do not worry with the name and do not worry with the notation. As long as you can make the Punnett Square properly or the probability rules properly, you should be in a good position to do these crosses, right? All right, so now let's open up for some burning questions and curious thoughts, starting from Alok. He is waiting from a long time. Alok, please go ahead.
Okay. Hello, sir. So I wanted to ask you is that there are two parents who are homozygous, who are heterozygous. How is
Parents
it possible?
homozygous or heterozygous? What you are-
Heterozygous.
Mhm.
How is it possible for both of for them to have all the children, and let's say they have more than 2 children, to have all the children homozygous recessive?
I think it's very confusing question what you are asking, and a lot of things which you are changing the statement.
Okay, sorry.
Alok,
There are 2 parents who are heterozygous,
Alok,
and they have more than 2 children.
Listen, you have to be clear. First of all, you can just type your question clearly in the chat. We can look into that. Okay, so that we can save the time. A lot of people are here.
Okay, I'm getting mixed up in words. So
do not worry. Please type clearly so that we can then get back quickly. Yeah, we will talk to you. Hriday, you are next. Unmute and talk.
Yes, sir. So in a case where both of the parents, human parents, are heterozygous, in probability of children being albino should be independent of each other, right?
Well, see, if both of them are heterozygous, so it means they are carrier for that property in some way, right? Because they are not showing the trait, but they are actually having the possibility for that recessive trait to carry. And that's what we saw like in a capital A, small a case, right? So if I just go back to that slide right. So in this case, actually, they appear normal, both of them, because they are not having albino trait, but they are not capital A, right? So they are not completely normal. They have this one recessive property. So based on this, like, it is possible that 1 children out of 4 or 25% probability that one of the children may be albino. But it just be like if all the 4 children show up and then only one of them shows the albino trait. Now, you can rephrase your question based on this idea, what we have just
So in case there are 4 different, all the children are from different pregnancies, then the probability of one being albino is much less than 25%, right? Is not 25% or even close to it, right?
You're right. Like, in each time, every time when the pregnancy happens, there will be 25% probability. Correct. Like with each of these gamete being showed up, there will be possibilities of only 25% each time that the children might be albino, or it may be normal completely, or maybe carrier. So both ways are possible, yes. But it will be every time, yes, 25%. All right, Karthik, you are next.
Yes, sir. I had a doubt that the blood donation. So what is the conditions behind it? Like rhesus factor and all that.
Yeah, so I think let's not go in detail of blood donation right now, because what we are looking at only blood grouping, which governs with the genetic rules when we are looking at the crossing and the properties. For the blood donation, you are looking at separately the the blood is separated, the corpuscles are separated, the platelets are being separated, and then depending on the again, you need to know which donor is going to give for be used for which type of properties. Like, sometime, especially when you have, let's say, dengue fever, you're only being given platelets, right? In some cases, you are going to give in the RBCs. So based on that, then you are separating these particles and those are being given, but that itself is quite detailed idea, which requires a lot of different type of.
So no, I'm just asking which blood group can donate to which blood group. How do we decide that?
All right, well, then now you have these tables yourself, and you can try to look into this, like which are the universal donors, which are the universal acceptors, which can be used for crossing each other. Like, all of these informations are here, and some of these are going to be questions for us as well in the quiz.
So, but will the RH factor cause any changes in this?
RH factor will not cause they change for this acceptance. But it should be positive or negative when we talk. That is a separate protein which is present on the RBCs, rhesus factor protein. but that positive can be separately matched. However, the main ABO with the universal acceptor and donor will only rely on these antigens, which is AB or none. Okay.
Okay, sir.
All right, Jayank.
Yes, sir. The yellow is the dominant factor in the pea plant. Then why we see only green plants around us, green pea plants?
You mean here, like, the seeds of this or the flower of this or which? Here, right? So green is dominant, right? So when we talked here, like we are looking at one dominant property, one recessive property, recessive trait. So pot color green, that's what I think you meant, right? Like what you see the pea pods. Is that what you want to know? Jayant, can you unmute and ask yourself the same question again?
Yes, sir. The yellow is the dominant factor. Then
Where it says?
Here, yellow and the green. Yellow is the dominant, no?
You mean the seed, the seed,
Yes,
green
sir. Yes,
seed
sir.
and yellow seed, right?
Yes, sir. Then why most of the seeds are green? We see around us.
That's right. So I think you are right in some way, like, what we, when we are looking at properties here, like yellow versus green, if yellow is dominant, then why we see mainly the this green thing.
Yes,
but
sir.
also there is a lot of selection which happens over the time. While in the cross, when we do, like in the cross of these pea plants, yellow versus green is only going to show the color of yellow. But it is possible that with the breeding, which happens when this particular population has been further bred, and therefore what you see is most of the seeds coming from this only. Therefore, you see most of the pea pods are carrying the green seed only. However, there will be population when you will definitely see some yellow as well. But this is the blend of the formats when they do breeding. Some traits are being bred much more, and therefore you see those much more in your vegetables or fruits likewise. That cannot just define the whole genetics per se, that why you don't see that everywhere. Because what is not happening, like, just the open crosses, right? What is actually happening, the formats, they do the breeding with the restricted ways of crossing what properties they want to propagate further. All right. Kinjal, you can ask now.
Sir, if alleles are just a sequence of nucleotides, why aren't there more than two alleles? Like for the color of flower, there are just purple and white. Why can't there be more than that?
No, right. See, for example, let's think about one more natural example of our eye colors, right? So our eye colors could be black, brown, blue. Some of these colors are being observed, right? So for the coloration of the eye, there's only one gene which is responsible, which is going to have multiple allele forms. Same thing is what we saw in the previous examples in the plant as well. So this stretch of nucleotide sequence, which denotes one gene, that gene is responsible for the property of that type of property of that color, right? However, because of some changes in that gene sequence or nucleotide sequence, we do see there is possibilities of either the black or brown or blue appearing. So therefore, you cannot say like these are just the nucleotide sequences. Why they cannot be? There are of course many genes, which are there. There are many in the same chromosome. There are multiple genes, and they are looking for many traits. So the same gene sequence is going to be responsible for one gene trait. And therefore that nucleotide sequence is very specific or unique for that particular gene. And that's what by now we know in human, we have 20,500 genes, which are governing many of these traits. So just to sum up, we have only for this particular property, we have one gene, which is having one nucleotide sequence, but that one nucleotide sequence has some minor changes, which is coming from both the parents, father and mother. And therefore you see allelic variants of that particular gene. I hope that just clarifies this point.
So sir, is that possible that synthetically we can amend some of the nucleotides and change the color of?
We can. Synthetic biology is really looking at this, because now we have ability to synthesize the nucleotides. We can actually think about making these changes, but of course doing this in human is very difficult because of a lot of ethical and the difficulty of doing these experiments. However, in the different other model organism, people are trying to play up with the different gene sequences in synthetic biology. Okay, because you can ask your question next.
Sir, like, is it possible that a single plant has both like inflated and a constricted P?
There will be some possibility, because again, that 9:3:3:1 ratio or 3:1 ratio, what we have been talking, those are not always exactly same. Like there is some, 3.14:1, that kind of numbers also show up. So that's
No,
not
like different. There can be cases like 341 plants had that case, but I am talking about a single plant. Like, is it a possible like some P like P flowers come of different color and some of different color? Taking the Mendel as the standard, like I'm not talking about the codominance and all.
That's right. So looking at this trait, I don't think that will be a possibility. But many other things which if we add up, it could be happening in nature because of many other factors. Something which you
Oh.
mentioned as well.
Like a situation may happen that the enzymes of the recessive trait may start releasing.
Absolutely. All right. We have reached almost to 4:48 now, and still a lot of interesting hands are being raised. We are going to start the quiz now, which is going to be for 15 minutes. Quiz is going to be based on today's lecture, which will be not very difficult. We are hoping that all of you who have paid attention to today's lecture and at least have, listened the points carefully should be able to perform very well. If you have any specific still question, I think I highly recommend you to also utilize the Google Forms well, because it looks like many of you have some very interesting questions and ideas which we could take forward and try to give you some more information at length. But given the short time which we have, and as soon as we start getting late, then all of you will start cribbing that you have to go for next class. So we can't exceed the time too much as well. So therefore, I now request all of you to join the quiz in Moodle. Some of you we could not answer you on time, given the shortage of time, please do put in the Google form or directly email me. We'll be making sure to give back to you the right answers and details to clarify your doubts. Nevertheless, I think given the class timing, we are going to stop now, and we are going to start the quiz, which will be in Moodle like earlier. And once you finish your quiz, you can leave. Please do read the genetics chapters much more closely and observe them in much more detail, because you will see, we are going to have many interesting observations and questions from this area. And the next lecture, which will be also based on genetics only, but the model organism is going to be changed, where we are going to talk about from Mendelian genetics to the Morgan's genetics, and moving from pea plants to the fruit fly or Drosophila melanogaster. So we will meet in the next class, and I'm going to release the lecture video beforehand shortly after this quiz will be done at around 5:30 p.m. or so. So start watching the next lecture. Be prepared beforehand. The 7th lecture will be my last lecture of my part of the MCV module. And then you can ask any clarity if you need for any questions in the Google Forms, which you'd like to take forward. And again, next class, I'll be live available to talk to you and give you some more insight. Thank you very much, everyone. And you can go ahead and do the quiz. Good luck.